David Spaulding, and my advisor is Dr. Boriana Marincheva, Fishing for Viral Genes Involved in Protein Synthesis, a Bioinformatics Approach. Fishing is a nostalgic activity familiar to most, but in this project we use strange bait. Here we see a short segment of DNA which actually represents a particular protein product, used as a probe seeking similar viral protein sequences. Most people have some concept of what a virus is. When I think of a virus, I think of something point-like and distasteful. But viruses have a structure. A small amount of DNA with possibly a few small molecules is packaged in a protective protein coat. Protein synthesis is a little less well known, but all living things employ it. When you look at a person, what you can directly see is made of protein. Genetic information conveyed by RNA passes through complicated cellular machinery and is used to direct the synthesis of linear protein polypeptides. The process is also called translation. When it comes to bioinformatics, almost no one outside the field knows anything at all about it. In this study, we use a process called T-BLAST-N, which compares a protein sequence known as a probe sequence with all known genetic sequences which could generate it. Recently, the scope of known genetic sequences has increased to include a large number of viral genomes. The question posed by this study is whether we can use selected bacterial and human translation factors as probes to uncover unexpected similarities with viral protein products. Such similarities, termed homologies, would be unexpected and would warrant further investigation if uncovered. As you can see, the process we use is indeed quite a bit like actual fishing. Each step in real-world fishing has an analog in the methodology of this work. Recognizing that this diagram is somewhat more confusing than I intended, I located a short movie on protein synthesis. Here is a single part of the cellular machinery involved in protein synthesis. It is a transfer RNA molecule with an attached methionine amino acid. You will see the tRNA bind to M, or messenger RNA, along with the small and large ribosomal subunits to assemble a protein synthesis machine, or simply ribosome. Don't worry about the details, just get an idea of what's happening. Here we see an initiator tRNA molecule bearing a methionine residue bound to a linear strand of messenger RNA with a small ribosomal subunit. Here comes the large ribosomal subunit. Please bear in mind that this depiction is a gross oversimplification, as the large and small ribosomal subunits are composed of scores of individual protein and RNA molecules. To even get to this point, other factors, known as initiation factors, must come into play. Once initiated, the residue-by-residue elongation of the nascent protein is supported by elongation factors. Protein synthesis is one of the processes that must be subverted by a virus during an infection event. As the DNA inside a virus particle is nowhere near long enough to code for all of the molecules involved in a ribosome machine, we theorize that what is present in the virus must be sufficient to divert the machinery to the viral purpose. At the end of translation, other factors, known appropriately as termination or release factors, are required for the disassembly of the translation complex. Here comes one now. To summarize the overview of our study, we take the protein sequences of normal human and bacterial translation factors and use them as probes into viral genomes, looking for a high degree of similarity. While similar, enough of a difference might exist which could account for the virus's ability to subvert the normal course of protein synthesis. This difference might be exploited in a search for new therapeutic drug agents. I've said that T-blast-N identifies protein homologs. We need a discussion on how that identification occurs. As an analogy, it's somewhat like a police lineup through a one-way mirror, but here instead of a single suspect being ID'd by witnesses, we place one suspect on the lighted side of the mirror and run the entire set of 5 billion human beings on the other side, the dark side, each evaluating their relatedness to the suspect. They then exit the viewing room, having first lined up in order from closest relative out first to most distant relative out last. Oh, and each prominently displays a scorecard over their head reporting their relatedness to the suspect. Now back to probe proteins and homologs. Here we have a human gene coding for a human protein being used as a probe, which is going to be compared against all viral genetic sequences coding for viral proteins, which are scanned for scoring as candidate homologs. How is this implemented? Scoring starts by locating anchor sequences. Between probe sequence and virus sequence, the viral sequence will scan by the probe until an exact match of 3 or more amino acids is found. This work of pattern matching is what computers excel at. The search now casts to the left and right, hoping to simultaneously extend the match from the anchor point and improve the score. Scoring is a bit more complicated than simply lining up perfectly matching peptides. Peptides that are chemically similar still score positively, and there is a penalty for each gap. The alignment of chemically similar amino acids is highlighted here in pink. There are 2 ways a search can be terminated. First, the size of the probe sequence can be exceeded. Second, as the number of gaps increases or the quality of matches decreases to either side of the anchor, the cumulative score falls. Eventually, it reaches a minimum threshold which terminates the search. In a real T-blast-N search, the casting about to either side of the anchor could be much more extensive than depicted here. Settings for the width used to identify an anchor, along with a scoring matrix for chemical similarity and minimum termination threshold, are all selectable. To summarize the process, prokaryotic and eukaryotic translation factors were identified, and their corresponding protein sequences were evaluated and recorded. These protein sequences were then presented to T-blast-N, returning a list of homologs. The best scoring hits with a score less than 0.01 were segregated from those of lower quality, score greater than 0.01. Reports were generated by aligning the returned homolog sequences along with the probes which located them. The above steps were then repeated using a new factor from a list of factors to be investigated. In the analysis phase, collation by probe, collation by virus, and collation by viral host clade were performed. Inspection of the collations raised more questions, directing further investigation. I just mentioned that the homologs are cataloged, and here are some example entries from that file known as the protein matrix. Information used for every step is preserved here, along with all intermediate results. Here we have an alignment report for eukaryotic release factor 1, listing 3 hits below the probe used to identify them. As long as gaps, indicated with red hyphens, are not too numerous, they do not seriously impact the quality of alignment. Also note the asterisk, colon, and periods under each of the 4 lines of alignment. Asterisks denote perfect alignment. Examples are highlighted in red at the bottom. Colons and periods, highlighted in pink and purple at the bottom, denote less than perfect alignment. The Clustal W program assigns partial credit anyway, according to the scoring matrix. Clustal W is the source for all the highlighting you see in this presentation. Here is the homolog count segregated by the significance threshold of 0.01. We found 18 high significance homologs with an additional 37 homologs of lower significance. Earlier I mentioned that the analysis involved collation. Here the sort is by translation factor in the yellow left column. The number of homologs in each grouping is reported in the next slide. Here's a list of translation factors used to date and a count of their homologs, bacterial and human. Here's the second collation. Homologs are now grouped by the virus coding for them in the yellow left column. In the right red column are the various probe factors for which homologs were found in each virus. Two results of note. The topmost group lists a virus coding for 3 homologs, the most per virus yet. 2 viruses list homologs for release factors 1 and 2. This was a trend for bacteriophages. What caught our attention was that mimivirus had homologs to both prokaryotic and eukaryotic translation factors, indicated in red. This is a portion of the third collation, which is the sort by the viral host clade. This showed that many viruses infecting rodents coded for homologs to release factor 3, indicated in red. There's been no follow-up yet to this observation. Here's a diagrammatic picture of mimivirus, the one with 3 hits. Mimivirus is a virus infecting an amoeba, which is common in soil. It has the largest known viral genome, in excess of 1.2 million base pairs. Here's the alignment between the probe eukaryotic release factor 1 with its mimivirus homolog. The alignment only includes a subset of the probe ERF1 sequence, with unmatched residues on either end indicated in red. Note that there are a few gaps within the alignment, 2 of which are only 1 peptide long, and another is 46 peptides long. As mentioned previously, these gaps are relatively unimportant as long as they leave long blocks of continuous matches. Here are the exact matches in red, and add the near matches in other colors. This was the best scoring hit in the study to date. When considering the fact that mimivirus shows homology to both eukaryotic and prokaryotic factors, a ready explanation comes from the life habit of the host amoeba, a eukaryote, which consumes and digests prokaryotic soil bacteria intracellularly. This animation is meant to illustrate that mimivirus could inherit eukaryotic genes from the host and prokaryotic genes from the bacteria. Further work includes processing the numerous additional translation factors already identified. Large homology sets have been identified for release factor 1 and release factor 2, and phylogenies should be generated for at least these factors. Additional work is needed on the mimivirus theory of horizontal gene transfer. And finally, during the readings prior to commencement of this work, many computer automation tools were discovered. But due to the highly repetitious nature of the work, implementation of these tools is desirable to avoid wear and tear on the human operator. Otherwise, they begin to quickly feel like this. I would like to thank Dr. Marincheva and the ATP Summer Grant Program, along with the other individuals listed, for their support during this research.